imagine for a second, you have two completely identical jigsaw puzzles.
Okay.
Like same piece shapes, same picture of, I don't know, beautiful landscape. You pour all the pieces onto a table. If you want to put them together, you intuitively group them by what is actually on the pieces.
Yeah.
Right? Like the blue sky pieces go in one pile, the green grass pieces go in another.
Yeah, exactly. You're looking for the fundamental truth of the image, to organize your workflow.
Right. But let's add a twist here. What if one of those puzzles was printed in a factory with slightly darker ink?
Oh, I see where this is going.
Yeah. If you take those mixed pieces and hand them to a traditional computer algorithm, asking it to sort the pile, it ignores the sky. It totally ignores the grass. It just groups all the dark ink pieces into one pile and all the light ink pieces into another.
Right. The algorithm becomes completely blind to the actual picture because a manufacturing quirk it hijacked its attention.
Exactly. And that dynamic is, it's the single largest bottleneck in modern biology today.
Okay.
And decoding that is our mission for you on this deep dive.
It really is a massive hurdle.
So scientists are generating these massive data sets of single-cell information. They're sequencing the biological makeup of individual cells to find disease markers or understand how tissues grow. But when they try to combine data from different patients or different laboratories or even samples run on different days, they hit this wall called batch effects.
Yeah, the dark ink, basically.
Right, the dark ink. So today, we are analyzing a really groundbreaking software package called Coralysis. And this completely re-engineers how we solve this problem.
And, to really understand the magnitude of this problem, we first have to look at what single-cell sequencing actually is.
Right.
We aren't just taking a piece of tissue and tossing it in a blender to get, an average reading. We are getting a high-definition mathematical profile of every single cell. It is an absolute ocean of data.
Which is incredible, but also super messy.
Extremely messy, because the moment you try to merge a data set from a lab in, say, London with a data set from a lab in Tokyo, those are our batches, by the way, the technical noise just overpowers the biology.
So like minor lab differences.
Exactly. The London machine might run half a degree warmer. The Tokyo technician might use a slightly different chemical reagent. That technical noise is the darker ink.
So the algorithm just groups all the London cells together and all the Tokyo cells together instead of grouping the actual heart cells with the heart cells.
Precisely.
Well, before we get into how Coralysis actually circumvents this, we need to trace the failure points of the current industry standards, because our sources highlight heavyweights like Seurat, SCVI, and Harmony. Why do these incredibly sophisticated algorithms fall for the dark ink?
The failure basically stems from their foundational approach to data integration. These current methods, they rely on constructing a joint low-dimensional space.
Okay, that sounds intense. What does that mean in practice?
Right. It's a fancy way of saying they take two massive data sets, squash them down to their most prominent features, and aggressively look for mutual nearest neighbor.
Mutual nearest neighbors.
Yeah, so they scan dataset A and dataset B, find cells that look somewhat similar, and just computationally force them together to anchor the two datasets.
That makes intuitive sense if you're comparing two identical healthy tissue samples, right?
Sure, in a perfect world.
But what happens in a scenario where the data sets are completely imbalanced? Say dataset A contains a highly specific ultra-rare immune cell because the patient was fighting off an infection.
Yeah.
But dataset B is from a totally healthy patient and doesn't contain that cell at all.
Right.
If the algorithm is mandated to find nearest neighbors, isn't it going to force a connection that just doesn't biologically exist?
It creates an absolute data car crash. The sources actually refer to this as the imbalance trap.
The imbalance trap?
Yeah, because algorithms like Harmony are just so desperate to align the batches and make the final graph look uniform, they will merge transcriptionally similar but fundamentally distinct cell populations.
Oh, wow. So they just blur the lines.
Completely. We can look at a specific case study from the data involving immune cells. When SCVI and Harmony attempted to integrate resting immune cells with interferon-stimulated cells, meaning cells actively fighting a threat, they completely destroyed the biological lines.
What did they merge?
They computationally mashed CD14 monocytes together with CD16 monocytes.
Which are totally different.
Very different. And they took naive T cells, which have, never seen a pathogen, and merged them with memory T cells.
Wait. Memory T cells carry the specific biochemical record of past infections, right?
Exactly. They mashed a blank slate cell with a highly experienced cell just to make the math look pretty.
Okay, let's pause and lock in a quick checkpoint for you listening, because, the stakes here are massive.
They really are.
So, First, we established that batch effects are technical artifacts like lab temperature or reagent differences that trick algorithms into sorting cells by their origin rather than their actual biology. Second, we saw that traditional algorithms try to fix this by forcing datasets to overlap, looking for those mutual nearest neighbors. And third, the fatal flaw. When datasets contain different populations of cells, this forced overlap destroys the underlying biological truth by merging completely distinct cell types.
And, the downstream consequences of that are severe.
Because the researchers are trusting this data.
Exactly. If a researcher's analyzing that flawed data, trying to isolate, say, a drug target unique to a CD16 monocyte, but the algorithm has secretly blended it with CD14 data just to make the integration look nice, the resulting drug development is based on a phantom cell.
A cell that doesn't actually exist in nature.
Right. You're building an entire research pipeline on a cracked foundation.
Which brings us to the architecture of Coralysis. This is an R-based software package designed to perform multi-level integration of single-cell data. It completely abandons that mutual nearest neighbors approach.
It does.
It uses a new engine called Iterative Clustering Projection, or ICP. So let's get under the hood here. How does ICP actually begin organizing this chaos?
Well, it fundamentally changes the sequence of operations. Instead of throwing all the raw data from all the batches into a giant mixing bowl and trying to untangle it, Coralysis builds a batch-balanced training set.
Okay, but how do you mathematically balance a data set if you have, say, 10,000 common T cells and only 5 rare dendritic cells? Won't the algorithm just be totally overwhelmed by the sheer volume of the T cells?
See, that is exactly the trap Coralysis avoids. It doesn't use the raw cell counts for the initial training.
What does it use?
It groups the cells within their original batches first and calculates the mathematical average of their features. By using these aggregated mathematical profiles rather than the individual cells, it creates a separate, perfectly leveled playing field.
Oh, I see.
Yeah, so a cluster of 5 rare cells gets the exact same initial computational weight as a cluster of 10,000 common cells.
That's brilliant. The algorithm is basically preventing the loudest voices in the room from drowning out the quiet ones.
Exactly.
So once it has this balanced training set, the sources describe a top-down divisive sorting mechanism. And I want to try visualizing this if I have a massive pile of unorganized laundry.
Okay, I like this analogy.
Right. And I try to sort it into 50 different, highly specific piles all at once, like angle socks, dress shirts, darks, lights, delicates, wools. I am practically guaranteed to misplace items. I'll throw a delicate dark shirt in the tough dark pile by accident.
For sure. It's too many variables to juggle at once.
Right. So Coralysis avoids this by not trying to find 50 cell types at once.
Exactly.
Right?
Top-down divisive clustering means it starts with just 2 piles. It looks at the entire biological data set and identifies the single largest axis of variation, which statisticians call the first principal component, or PC1.
So sticking with the laundry, just looks at the massive pile and says, lights and darks. Everything goes into one of those 2 clusters first.
Yes. Then it takes the darks cluster and splits that in 2 based on the next major variation, maybe separating cotton from synthetic fabrics.
Got it.
So that gives you 4 clusters total. Then it splits those, giving you 8, then 16, continuing down the tree. It only ever has to make a binary choice at any given step.
Which drastically reduces the error rate compared to trying to find 50 clusters simultaneously.
Exactly.
To make those binary choices, the ICP engine relies on a mathematical formula called L1 regularized logistic regression. Let's dissect that because it sounds intimidating, but the mechanism is actually brilliant.
It is.
Logistic regression is just a statistical way of predicting the probability that an item belongs to a certain category. But what is the L1 regularization doing to that probability?
Well, regularization is a mathematical penalty applied to complex models to prevent them from overanalyzing noise. Think of L1 regularization as a highly aggressive editor.
Okay.
When Coralysis is trying to determine what separates those CD14 and CD16 monocytes we talked about, it is looking at a matrix of thousands upon thousands of gene expression levels.
Right, an ocean of data.
But most of those genes have absolutely nothing to do with the difference between those 2 cells. So L1 regularization actively penalizes the algorithm for using too many features. It literally shrinks the mathematical weights of irrelevant genes down to absolute 0.
Wow. So it deletes the distractions, it forces the model to ignore the background noise, and base its sorting decision entirely on the bare minimum number of core-defining genes.
Exactly. And it doesn't just guess and move on either. Coralysis constantly audits its own accuracy during this process using a metric called the adjusted RAND index, or ARI.
What exactly is an adjusted RAND index measuring in this specific context?
It's a measure of similarity between two data clusterings, adjusted for the probability of random chance.
Okay.
So the algorithm sorts the cells into groups, assigns a probability score to each cell, and then it runs the sorting process again. The ARI compares the first attempt to the second attempt.
So checking its own work.
Right. If the ARI score is low, it means cells are jumping back and forth between clusters. The model hasn't found the true biological boundaries yet, so it iteratively trains, adjusting its parameters, until the ARI score approaches 1, meaning the clusters are completely stable and the cells stop moving.
And the mechanism driving those specific splits is based on the batch-wise median of maximum assignment probabilities. It basically finds the median confidence level for each batch and uses that as a threshold to split the trait.
That's right.
Okay, let's do another quick checkpoint for the listener to consolidate this algorithmic deep dive.
Good idea.
We've covered 3 massive innovations Coralysis brings to the table. Number 1, it prevents rare cells from being ignored by creating a mathematically averaged batch-balanced training set.
Yep.
Number 2, it avoids massive sorting errors by using a top-down binary split method, dividing the data step by step rather than all at once. And number 3, it isolates the true biological signal by using L1 regularization to aggressively zero out irrelevant genes and verifies its work using that adjusted RAND index.
Having the theory laid out is one thing, but we have to look at how this performs against real messy biology. The sources provide a benchmark case using peripheral blood mononuclear cells, commonly known as PDMCs.
Right, so the researchers took 2 separate single-cell RNA datasets. One batch contained 8,276 cells, and the other batch was roughly half the size, with 4,770 cells.
A classic imbalanced scenario.
Exactly, where traditional methods usually force bad overlaps. How did Coralysis handle it?
It navigated the imbalance perfectly. It successfully separated the B memory cells from the B naive cells, and more importantly, it accurately grouped the CD16 plus monocytes across the batches, even though one batch had a drastically different proportion of them.
So it didn't blur them into the more common cell types just to balance the final visualization.
Not at all.
The phrase the sources use for this is prioritizing bioconservation over batch correction. Algorithms like Harmony prioritize batch correction. They want the integrated data to look perfectly blended, even if they have to destroy subtle biological variations to achieve it. Coralysis refuses to make that compromise. It protects the biological truth.
And, that bioconservation extends far beyond just analyzing RNA. If Coralysis is genuinely isolating fundamental biological signals, the math really shouldn't care what type of biological data you feed it.
True.
And the sources detail its application in single-cell proteomics, specifically using ADT and CyTOF data.
Let's clarify those modalities real quick. RNA sequencing, which we just discussed, is transcriptomics. You are measuring thousands of genes actively being transcribed inside the cell. Proteomics looks at the proteins on the outside of the cell.
Exactly.
And ADT stands for antibody-derived tags, which allows researchers to measure specific surface proteins alongside the RNA. CyTOF is cytometry by time of flight, where researchers attach heavy metal isotopes to antibodies to measure proteins without the visual overlap issues you normally get with traditional fluorescent tags.
That's a great summary. But the crucial detail here is the feature space. With RNA, Coralysis is analyzing a matrix of maybe 20,000 genes. But in the CyTOF whole blood dataset analyzed in our sources, there were only 39 proteins measured per cell.
Wait, 39? That is an incredibly sparse matrix. How can the algorithm accurately cluster distinct cell populations with only 39 data points per cell?
It leverages the exact same ICP engine we discussed earlier. The researchers took a CyTOF dataset combining healthy patients and patients suffering from an H1N1 influenza infection.
Okay.
Despite the fact that the algorithm only had 39 parameters to look at, it successfully integrated the 2 batches. It was so precise that it identified an ultra-rare population of basophils that constituted merely 0.5% of the total cells in the dataset.
Finding a 0.5% cell population using only 39 clues is just staggering. And that precise targeting capability allows Coralysis to execute another major function, reference mapping.
Yes. Reference mapping is how we actually scale biological research. Instead of manually annotating and identifying cell types from scratch every time a lab runs a new experiment, researchers can take their new unlabeled data, called the query dataset, and project it onto an existing pre-labeled reference map using the trained Coralysis models.
I have a fundamental question about the integrity of that process, though. Does the biological state of the reference map dictate the results? Like, what happens if I take a batch of highly stimulated virus-infected cells and use that as my foundational reference to map a batch of perfectly healthy resting cells?
That's a great question. The sources include a fascinating case study on that exact scenario, and it actually exposes a vulnerability in the L1 regularization we praised earlier.
Oh, really?
Yeah. When researchers used resting healthy immune cells as the reference map to analyze stimulated cells, the accuracy remained remarkably high. But when they reversed it, using the actively responding stimulated cells as the reference map for the healthy cells, the accuracy completely plummeted.
What caused the model to break in reverse?
The L1 regularization acted as too aggressive of an editor. When building the reference map from the stimulated cells, the algorithm latched onto a specific gene called CCL8 to define what a monocyte was.
Okay, what is CCL8?
The biological reality is that CCL8 is a chemokine gene that is severely upregulated only when a monocyte is actively responding to an immune threat. It is virtually non-existent in a healthy resting monocyte.
Ah, I see the trap. The algorithm defined a monocyte purely by the presence of a panic button gene. So when it scanned the healthy query data and didn't see the panic button, it concluded there were no monocytes present at all. It basically overfit the model to a transient state.
Precisely. To solve this, the researchers had to change the mathematical penalty. They switched the Coralysis algorithm from L1 regularization to L2 regularization.
Let's compare those mechanisms. We established L1 shrinks the weights of irrelevant features to absolute zero. How does L2 change the math?
Well, instead of penalizing the absolute value of the weights like L1 does, L2 regularization penalizes the squared magnitude of the weights. Practically speaking, this means L2 shrinks the influence of less important genes, so they're very small, but it almost never reduces them to absolute zero.
Okay.
It retains all the coefficients, just minimizing the noisy ones.
So if L1 is a highly focused laser pointer, illuminating only the single most critical gene, L2 is more like a floodlight. It captures the broader context of the cell's entire genetic profile, which gives the model the flexibility it needs when comparing a sick cell to a healthy one.
That's a perfect way to put it. And that structural change fixed the reference mapping issue entirely. The L2 floodlight allowed the algorithm to recognize the healthy monocytes based on their broader protein signatures, even without that CCL8 gene present.
Awesome. Time for our final checkpoint.
Let's hear it.
So we expanded Coralysis beyond RNA into proteomics, proving it can find rare cells like a 0.5% basophil population using only 39 surface proteins. We explored reference mapping, which projects new data onto existing atlases, and we uncovered a critical caveat. When mapping healthy cells onto a diseased reference, you must use the L2 regularization floodlight instead of the L1 laser pointer to avoid overfitting to stress response genes like CCL8.
Right. Which brings us to a phenomenon in the data that completely redefines how we interpret computational output.
I'm right.
Throughout this entire process, Coralysis assigns a cluster probability score to every single cell. It mathematically states how confident it is that a cell belongs to cluster A versus cluster B.
Logically, if the algorithm assigns a cell a very low probability score, say it's only 45% confident, my assumption is that the algorithm is failing, like the data is too noisy or the parameters are wrong and the math is just confused.
And that is the standard assumption in data science. But the ultimate revelation from our sources is that this mathematical probability actually mirrors physical, real-world biology.
Wait, really?
Yeah. A low probability score doesn't mean the algorithm is broken. It means the physical cell itself is in a transient state. It is actively transforming. Conversely, a high probability score near 100% means the cell is in a steady state or terminally differentiated.
So the mathematical confusion is a literal map of a cell's biological evolution.
Exactly. We can observe this perfectly in the human embryoid body data. An embryoid body is an in vitro cluster of pluripotent stem cells that are actively developing into the 3 primary germ layers of the human body.
So it's an environment defined by rapid cellular transformation.
Exactly. When Coralysis analyzed this data set, the terminally differentiated cells, the ones that had locked into their final identities like neural crest cells or endoderm 1 cells, received incredibly high probability scores. The algorithm knew exactly what they were because the cells knew what they were.
But the stem cells and the intermediate cells that were halfway through the process of specializing caused the probability scores to drop.
Yes. For example, early-stage mesoderm cells, which haven't yet committed to becoming bone or muscle or blood, registered very low probability scores. The algorithm was capturing the biological reality that these cells existed on a gradient between two distinct identities.
And the researchers utilized these probability scores to actively discover new developmental mechanics.
Yeah, they did. By taking the cells and organizing them purely by their Coralysis probability scores, from the lowest confidence to the highest confidence, they could track the exact genes that activated as a cell matured.
That is so cool.
They isolated those early-stage, low-probability mesoderm cells and found a heavy concentration of activity in a gene called TMM88.
What role does TMM88 play in that transient state?
TMM88 is a known inhibitor of WANT signaling. The WANT signaling pathway is, well, it's a complex network of proteins that essentially dictates cell fate and tissue regeneration.
Okay.
By expressing TMM88 and inhibiting that WANT signal, these transient mesoderm cells undergo the crucial biological shift required to commit to becoming cardiomyocytes, which are beating heart muscle cells.
Wow. By simply looking at the exact coordinates where the math was the most uncertain, researchers located the genetic switch that commands a stem cell to build a heart. That completely flips the paradigm of how we handle noisy data.
It really does. We have spent decades trying to force algorithms to give us clean, distinct categories. We wanted black and white answers. Coralysis proves that the gray areas we've been trying to correct out of our data sets are actually holding the most critical biological secrets.
Which brings us to the ultimate takeaway for you, the listener. We started with the massive bottleneck of batch effects, where traditional algorithms destroy biological truth by forcing distinct cells together just to make the data look uniform.
Right.
But Coralysis offers a fundamentally different architecture. It uses batch-balanced training sets to protect rare cells. It uses top-down divisive clustering to avoid sorting errors. It uses L1 and L2 regularized regression to filter out the noise. And it operates across transcriptomics, proteomics, and reference mapping.
It essentially provides the computational infrastructure to integrate millions of single cells from across the globe without sacrificing the subtle realities of human biology.
And that reality leaves us with a final, really provocative question for you to mull over. If a purely mathematical algorithm's confidence score perfectly mirrors how mature or transient a physical cell is, what does that mean for our fundamental definition of a cell type?
That's the big question.
For centuries, biology has categorized cells into neat, distinct buckets: a T cell, a monocyte, a neuron. But if the data is showing us that cells exist in constant states of transformation, are there truly distinct, hard-coded cell types in our bodies? Or is human biology just one continuous, highly fluid gradient of probabilities?
It's fascinating to think about.
When you stop forcing the puzzle pieces to match based on the ink, the picture that emerges is far more dynamic than we ever imagined. Thank you for joining us on this deep dive into the source material. Catch you on the next one.